Welcome back. So you are now familiar with what prokaryotic and eukaryotic cells are. So let's kind of set our prokaryotes aside and really dig into exploring what is in a eukaryotic cell. So eukaryotic cells are typically structurally complex. When you look at an animal cell or a plant cell, you see a lot of different things in it. You see things like a plasma membrane, nucleus, several different organelles, things like the Golgi apparatus and the mitochondria, and the cytosol. And around the cytosol and all of these components is a cytoskeleton, which is proteins that give it its shape like a skeleton. Then outside of the cell, you'll see a cell wall, a rigid structure around plants and fungal cells, or an extracellular matrix, a more pliable, sugary extracellular component in plant cell or in animal cells. So a plasma membrane surrounds all cells. No matter what kind of cell it is, all cells have a plasma membrane around it. That helps make sure that the things that are supposed to be inside of the cell are kept inside of the cell and don't just leak out. Plasma membranes are made of lipids. The main component is phospholipids, although there are other types of lipids that we'll talk about this semester. And there are membrane proteins. In this picture, these blue areas are membrane proteins. You can see there's different types of proteins here. You can see this phospholipid, which is kind of this red-yellow area, is divided into two layers. So there's a layer here and there's a layer here. So it is called a bilayer because there are two layers there. Each phospholipid molecule, if you look at just one of these, you can see it has a little head and then two little squiggly lines down it. So it has this hydrophilic head and these two little hydrophobic tails. So that hydrophilic head means it's able to associate with water. So that hydrophilic area turns towards the water. It'll interact with the water and other polar and ionic molecules. These hydrophobic tails, because they are because these hydrophobic tails are nonpolar, they're not going to interact with water. So they turn inwards into the middle of the cell membrane so that it will not be interacting with water. It's interacting with the other nonpolar tails. So that means that a phospholipid molecule is amphipathic. It is both hydrophilic and hydrophobic. And it forms this lipid bilayer automatically when those hydrophilic heads interact outward and the hydrophobic tails turn themselves inward. Membrane proteins can also be amphipathic. So like this one has this hydrophobic ring structure here and hydrophilic extracellular and intracellular areas. This would be a amphipathic protein with a hydrophobic area, a hydrophilic area, and a hydrophilic area. And some of these membrane proteins have sugars attached to them. That's what these little green spots are. These are carbohydrates. That makes them a glycoprotein. Glyco referring to the sugars on them, and protein referring to the protein of it. Adding those sugars can be important in signaling and allowing cells to recognize with each other and interact with each other. Proteins in the plasma membrane will have a variety of different roles. We're going to come back and talk about these when we come back to chapter 7. These plasma membrane proteins could include enzymes. Enzymes could do things to catalyze reactions, such as creating the cell walls. Some are anchors, so they are going to help anchor components to the cytoskeleton. Some are transport proteins. They move things across the membrane into or out of the cell. Some are receptors, so receptors that pick up extracellular signals so that the cell is able to tell, here's a hormone and then respond in kind, or pick up other signals, growth factors or something. Transport proteins and receptors will be transmembrane proteins. That means that their structure means that they're going to need to have an extracellular side that can interact outside the cell. They'll need to have an intracellular side that can interact inside the cell. So they're going to have to go all the way through the membrane. So they're going to be transmembrane proteins. Next, we have the nucleus. The nucleus is a very prominent structure. If you were to just, if this was your first biology class and you were looking at like an onion cell or a fish cell or something, if you're looking at something in the microscope, The nucleus would probably be one of the first things that you'd be able to see because it is so prominent. Within the nucleus is the DNA, and the nucleus is surrounded by a nuclear envelope. And the nuclear envelope has two bilayers. It has an inner membrane and an outer membrane. And within those membranes, there are pores. Pores are openings that allow things to come in and out of the nucleus. So you could send like a transcription factor into the nucleus, or you could, if your nucleus is making a piece of RNA, it can send that out of the nucleus. And those pores are transport channels that are lined with a nuclear pore complex. The number of chromosomes in the nucleus is a species-specific characteristic. That means all of one species should have the same number of chromosomes. Having more chromosomes does not mean you're more complex, or having less does not mean you're less complex. But it is just a specific characteristic for species. So for humans, you should know we all have 23 pairs of chromosomes. So that all humans should have 23 pairs of chromosomes. Chromosomes are going to be most easily visualized during mitosis, because during mitosis they condense down to be really nice and thick. During interphase, they're more dispersed. That dispersed phase of chromosomes is called chromatin. It's kind of opened up a little bit easier for the DNA to be accessible to the cell. And because it's a little bit more diffuse, it's a little bit harder to visualize. There's also nucleoli in the nucleus. You will see that word pronounced a lot of different ways, but nucleoli or nucleolus. This is where ribosomal RNA is being created. So within the nucleus, we're making lots of RNA. Ribosomal RNA is really important because it is what creates the ribosomes to allow proteins to be made. And proteins are what allow your cell to function. So nucleoli are where we are creating ribosomal RNA. And that area of the chromosome, or that area of the nucleus, is a pretty specific spot where it is making that ribosomal RNA. Next, we have mitochondria. Mitochondria are found in all eukaryotic cells. Plants, animals, fungi, we all have mitochondria. This is where aerobic respiration happens. We are not going to go through aerobic respiration in this class. You may have memories of talking about glycolysis and Krebs cycle and electron transport chain from other cells. So Krebs cycle, electron transport chain, that happens in the mitochondria. That's what aerobic respiration is. Mitochondria are pretty similar in shape and size to bacteria. They look like tiny little bacteria in our cells. And most of our cells have hundreds of mitochondria. And mitochondria have an inner and an outer mitochondrial membrane. We have an inner membrane, first bilayer around them, that's actually pretty similar to bacterial membranes. And then an outer mitochondrial membrane that's a little bit more similar to our eukaryotic membranes. The inner mitochondrial membrane surrounds the matrix, which is that inner fluidy stuff inside of the mitochondria. The matrix also includes small circular DNA molecules. So mitochondria have their own DNA, which is small and circular, pretty similar to bacterial DNA. And mitochondrial DNA will encode RNAs needed to make proteins specific for the mitochondria, and also will make ribosomes in protein synthesis. The mitochondria has enzymes and intermediates needed to be able to oxidize sugars. That means we are going to take sugars, remove the electrons from them to create energy, and that energy is going to be created in the form of ATP, or adenosine triphosphate. So cells that have a really high energy need, such as sperm and muscle cells, things that are moving a lot, need a lot of energy, are going to have a lot of mitochondria. The chloroplast is an organelle where photosynthesis occurs. Chloroplasts are not found in animal cells because we eat. We don't do photosynthesis. But they are found in plants and algae. They can be really large, and they can be really numerous in the cells of green plants. They are responsible for a lot of the green color that plants have. And they're also surrounded by an inner and outer membranes. They have a series of flattened sacs called thylakoids, and they are stacked into grana. They are connected by stroma thylakoids. Oops. Chloroplasts are where photosynthesis takes place. Photosynthesis is a process of taking carbon dioxide out of the air, using the energy from sunlight to take those carbon dioxide molecules and combine them to produce sugars and other organic compounds. Although we don't do it, we are reliant on plants performing photosynthesis to be able to have sugars and organic compounds for us to eat. Chloroplasts are found in photosynthetic cells, and they have most of the enzymes needed for photosynthesis. The reactions in a chloroplast that depend on the sunshine are going to be occurring in or on the thylakoid membranes. So getting the energy from the sunlight to create ATP to have chemical energy in your cell. These reactions occur on the thylakoid membrane. Sometimes these are called the light reactions. Then the reactions that involve taking carbon dioxide and reducing it to sugar occur in the stroma, or the fluidy gel-like structure inside of the chloroplast. So the thylakoid is where the photo part takes place. The stroma is where the synthesis parts take place. Chloroplasts, like mitochondria, have their own ribosomes and small circular DNA. They also look a lot like little baby bacteria in our little cells. They have DNA that encodes RNAs and proteins that are needed for the chloroplast. There are also plastids. These are a class of plant organelles. Chloroplasts are one type of plastids. There's also other types. One would be chromoplasts. You can see this chromoplast has a lot of little like orangey red pigments in it. Chromoplasts have pigments in it that give plants a lot of their color. So when you look at a flower or a fruit and it has these beautiful bright colors, a lot of that is due to chromoplasts. Amyloplasts are plastids that are specialized to store starches. Another type of plastid that I really like is responsible for helping a cell figure out which way is up and down, so that it knows which way to send its roots and it knows which way to send its stem growing when that seed is germinating into a plant. Okay, so we've talked a little bit about how chromosomes, or how chloroplasts and mitochondria, these two organelles in eukaryotes, are really similar to bacteria. They are about the same size of bacteria. They're about the same shape of bacteria. They have double membranes around them, and that inner membrane is similar to bacteria. It has bacterial type lipids in it. Mitochondria and chloroplasts have circular DNA molecules. These circular DNA molecules do not have histones. Even though they are within a eukaryotic cell, the DNA within mitochondria and chloroplasts acts like it is a prokaryotic DNA. The ribosomal RNA, the sequences, the size of the ribosomes, these are similar to bacterial structures. Mitochondrial and chloroplast RNA are sensitive to inhibitors that affect RNA of bacteria. Protein synthesis in mitochondria is inhibited by drugs that inhibit bacterial protein synthesis. The type of protein factors used in protein synthesis are similar to bacterial proteins. Also, mitochondria and chloroplasts can divide on their own, in a similar structure to, or in a similar pattern to how binary fission occurs. They have their own DNA, messenger RNA, transfer RNA, and ribosomes. They really are bacteria, little bacteria that are now become dependent on our cells, and our cells have become dependent on them. We have this kind of symbiosis going on between these mitochondria and our cells now. So the endosymbiotic theory is a theory that proposes that mitochondria and chloroplasts actually were derived from bacteria. They were bacteria that were endocytosed and now have become symbionts of our cells. So the endosymbiotic theory suggests mitochondria and chloroplasts evolved from ancient bacteria and then established a symbiotic relationship with primitive nucleated cells. So a long, long time ago, our ancestor eukaryotic cell endocytosed a cell that was really good at making energy, and that became a mitochondria. And then later it happened again. Endocytosed a cell that was really good at doing photosynthesis, and that became a chloroplast. Kind of crazy, but all the evidence, this is what the evidence points to. There's two different theories here. There's an outside-in theory and an inside-out theory. In the outside-in theory, a proto-eukaryote, this is the ancestor of our eukaryotic cells, wasn't yet really a eukaryote, but it was kind of our ancestor. In this proto-eukaryote, the nuclear envelope formed by invaginating the plasma membrane. So the plasma membrane is coming in, forming a nuclear membrane. These cells developed the ability to endocytose, to do phagocytosis, to bring in nutrients and particles. And some of those particles that were brought in were bacteria. And instead of digesting these cells, it surrounded these cells by the host cell membrane and evolved. These evolved into eukaryotes right along with this proto-eukaryote. In the inside-out theory, one form of evidence that goes along with this is that archaeal cells sometimes form protrusions, these little blubby protrusions from the archaeal cell. So the proto-eukaryote could make these protrusions, which would allow it to interact with nearby bacteria. And these protrusions then enveloped the bacteria, and the bacteria then evolved into mitochondria. These protrusions also made it possible to form a primitive endoplasmic reticulum. And the nuclear membrane also originated from these outer plasma membrane protrusions. Endosymbiosis is not just something that we kind of hypothesize could have happened a long time ago. It's something that's still occurring today. So algae, dinoflagellates, diatoms, photosynthetic bacteria, all live as endosymbionts in the cells of more than 150 existing protists and invertebrates. And our ideas about how eukaryotic cells acquire their complex array of organelles does have to remain speculative because we are not able to directly test them. Okay, so that's kind of a crazy story to end on here. One of my favorite things to talk about in this class. We do have some more eukaryotic structures to talk about. So in our next video, we're going to talk about the plasma membrane, or we're going to talk about the endomembrane system, things like the Golgi and the endoplasmic reticulum. We'll talk about some vesicles and lysosomes. I'll see you then. Bye.